Associate Professor Kieran Harvey's research findings are important for understanding species diversity and development and are directly relevant to human diseases such as cancer. Organ size control is a fundamental aspect of biology and varies greatly among animals. Signalling networks that control organ size are only just beginning to be unravelled. Foremost among them is the recently discovered Hippo pathway. Greater knowledge of size control will potentially have a huge impact on human cancer and degenerative diseases and provide fertile ground for therapeutic interventions. Associate Professor Harvey was an integral member of the team that discovered the Hippo pathway and was the first to show that it is evolutionarily conserved and also that it is mutated in human cancer. More recently, his laboratory discovered that the Hippo pathway controls organ regeneration. Associate Professor Harvey, congratulations. I invite you to present your talk entitled Control of Organ Size by the Hippo Pathway.
Well, thank you very much, Marilyn and Suzanne, and to the Academy for awarding me the Gottschalk Medal. It's really a fantastic recognition, and especially given the previous winners that have won this prize. So I wanted to start with a quote from a prominent English naturalist, J.B.S. Haldane, who made the observation that the most obvious difference between the most obvious differences between different animals are differences of size, but for some reason zoologists have paid singularly little attention to them. And this observation really held true for the majority of the last century. Developmental biologists spent a lot of time understanding form and patterning, but didn't really interrogate size in any great detail. And this is surprising given the very obvious differences in size that we observe in the animal kingdom, even in specific clades such as mammals. So this was something that I wanted to address, and I wanted to do that by understanding or identifying genes that regulate the size of specific organs. We also realized that the genes that we discovered that would control organ size were also potential new cancer genes. And this was very important because at the time, many cancer genes were known to exist, but we didn't know their identity. And the new generation of genomic technologies that Georgia presented on yesterday weren't available. So we had to use different approaches to identify human disease genes, such as cancer genes. So the approach I took was something that was done in Ishwar Hariharan's laboratory, my postdoctoral supervisor, who actually was a former PhD student with both Suzanne and Jerry at the WEHI. So in Ishwar's lab at Mass General Hospital, we used the model organism Drosophila melanogaster to understand organ size control. And our approach was very simple. Basically, we fed flies a mutagen, and then I recovered flies that had tissue overgrowth. We then went on to map the lesion, the genetic lesion that gave rise to this phenotype, and studied how it controlled organ size. And in parallel with colleagues at Mass General Hospital, we went on to assess the mutation status of the human equivalents of these genes in cancer by sequencing them. And this was a very successful approach, and we identified a number of known cancer genes as well as novel growth control genes that were also involved in human cancer. Most notably, the pathway that I'll just talk to you about today that was discovered in Ishwar's lab is known now as the Hippo pathway. So on the left here, I've got a picture of a wild-type Drosophila eye, and on the right is one that harbours a hippo mutation, specifically in the eye and head. And what you can notice is that the hippo mutant eye is significantly larger than the wild-type eye, and it's very folded and convoluted to fit all of this extra tissue into the head. If we look at this phenotype at cellular resolution, what you can see is that unlike wild-type tissue, which is stained here in green, you can see a single cell layer in between each of the Drosophila unit eyes, whereas in a hippo mutant, this beautiful architecture is disrupted. And what we see is the introduction of many extra cells, which gives rise to this tissue overgrowth phenotype in a Hippo pathway mutant. So to summarise many years' work by myself and other important colleagues in Ishwar's lab, we identified 3 genes, which we termed Salvador, Warts and Hippo, which we showed that these genes work together to limit tissue size. We showed that they do this by controlling cell growth, or the size that cells become, as well as by regulating the production of new cells via proliferation and the maintenance of existing cells via survival or apoptosis, as we heard from Jerry this morning. And importantly, we also found that the human orthologs of these genes are mutated in particular cancers. So that was important from a disease perspective, but it also gave us the first inclination that this pathway was conserved and played a similar function not only in insects but also in mammals. So this project really opened many important questions, and these were perhaps two of the most outstanding questions were, how do these three proteins that sit in the cytoplasm of cells regulate tissue growth? We had absolutely no idea how they did this. We presumed that they regulated transcription in some way, but we didn't know how. And we also figured that they must communicate with the extracellular world in some way, probably by, like many other classical signaling pathways that had been identified at the time, by responding to diffusible ligands that bind to receptor proteins that transverse the cell membrane. And so this second question was something that I was particularly interested in and pursued when I started my laboratory in Melbourne at the Peter MacCallum Cancer Centre. And in our first year, we discovered the first transmembrane receptor for the Hippo pathway, which is known as the fat cadherin. And then my group and many other labs throughout the world in the last decade or so have identified now many different Hippo pathway proteins. We've discovered more than 40 that exist in Drosophila and also in mammals. So it's really a complex signaling network. But what's different about Hippo pathway signaling is that unlike many of the classical signaling pathways that we know about that are regulated by diffusible ligands predominantly and transmembrane receptor proteins, the Hippo pathway is different. We can summarize it on this slide. The Hippo pathway really seems to be controlled by fundamental cell biological properties of cells that belong to three-dimensional organs. So, for example, proteins that regulate apical basal polarity, or whether a cell knows whether it's up or down, planar cell polarity, or the orientation of cells within tissues, cell-cell adhesion, all regulate the Hippo pathway, as well as key regulators or key components of the cytoskeleton, such as actin and microtubules. And this is particularly interesting in the context of the evolution of the Hippo pathway. So I told you earlier that the pathway is conserved in insects and mammals, but work from many other groups, not our group, has shown that the Hippo pathway is really an ancient pathway, and core components of the Hippo pathway even predate Metazoan evolution. So it's sort of tantalizing to speculate that given the role of the Hippo pathway in responding to social cues between cells, that maybe it even had an important role in the process of evolution of Metazoans. So to summarize, we've used Drosophila genetics to a very powerful system to identify a novel signaling pathway that regulates organ size in many different species. We've shown that this pathway controls not only developmental tissue growth, but also tissue repair or regeneration in many different organs. And lastly, we've shown that the pathway is mutated in many different human cancers. So science is rarely an individual pursuit, and many people have made very important contributions to my career. My PhD supervisor, Sharad Kumar, my postdoctoral supervisor, Ishwar Hariharan, and important colleagues at Mass General Hospital, such as Nick Tapon, where this work really originated. I also want to acknowledge many fantastic people that have worked with me in my laboratory to really bring a richer understanding of the Hippo pathway and how it operates in different species. Also, key senior colleagues at Peter Mac for their support, as well as collaborators and sources of funding. And lastly, I want to acknowledge my family, who have given me great support throughout my life and have also shared many fine dining experiences with me. Thank you.
Thank you very much, Kieran. Phil.
Philip Kushel, University of Sydney. All organs are characterised by being surrounded by a fairly high tensile strength membrane. How does that feed back into your system of understanding?
Yeah, I didn't talk about this work today because it's not work that I've done, but something very exciting that's come out in the literature in the last few years is that the Hippo pathway really responds to mechanotransduction information. This is important from a developmental biology perspective, but also from a cancer perspective. So many solid tumours are stiffer in their elastic properties, etc., physical properties. And it's believed that could be a key driver of disruption of the Hippo pathway in solid tumours.
Jenny.
Jenny Graves, La Trobe. How tissue-specific are these Hippo pathways? I'm thinking of dogs with wrinkly skin. Do they also have overgrowth of tissue?
Yeah, it's interesting. One of the... we named Salvador after Salvador Dali because he said he would never die and these cells don't die. The competitor group that cloned this gene called it Sharpei because of the tissue folds. So that's sort of an aside. But tissue specificity, certainly in Drosophila, we know that the Hippo pathway controls the size of epithelial tissues, but also neural tissues. In mammals, we've also... people have shown that the Hippo pathway operates in most organs in our bodies and in mice. Muscle as well, skeletal muscle, heart muscle as well. So it appears to be a fairly broad regulator of organ size.
Please join me in thanking Kieran for an excellent talk.